So I'll say what's relevant. You see this little thing right here, this little line? This is a sheet of connective tissue that all the valves of the heart are carved out of. And what's wild about connective tissue is that it's not easy to get an electrical signal through there. So the way that the heart beats is it's this electrical current that moves through the heart. It starts up here at the top of the right atrium, what we call the SA node. And it's going to basically just spread through both atria, and then it's going to spread down to the ventricles. But the problem is getting it through that layer of connective tissue. It's what is referred to as an electrical blockage of the heart. And I bring this up because it's what happened to me. There was a time when I would start to exert myself. You know, normal heart rate is about 76 beats per minute. But if you start to exercise or run it up and down the stairs, your heart rate is going to start to increase just to keep circulation going to all the tissues that need the oxygen and to wash away the waste. But as my heart would start to get above 90 beats per minute, there's a blockage basically right here. So my atria and ventricles got thrown off rhythm. And it was almost like my heartbeat doubled instantly. So it would go from about 90 beats per minute to about 225 beats per minute. We had me on one of these little heart rate monitors. And what happens there is that how many times your heart beats is only part of the story. If your heart is beating that fast so quickly, it doesn't have a chance, the chambers don't fill up with blood before it contracts. So even though my heart rate was through the roof, my blood pressure, the circulation was slowing down. And I would get all lightheaded and pass out. And they were going to take my driver's license away from me if I didn't have heart surgery. So when I was 16, I had this heart surgery. They went up through my leg, and they basically burned off half of my SA node. So it prevented how fast my heart can beat. So I couldn't play things like football or basketball after that, probably why I got into golf. But, and also interested in how the heart works. So when we're talking about cardiac physiology, it's anything that you can make relevant, it's easier to remember. And so I know even though this isn't in our class, we all have hearts, it's all relevant to us. So it's worthwhile at least mentioning. And we'll see how it overlaps with some of the stuff from 1406. So just as a quick visual representation of what we're talking about, we've got the heart, and I even have a heart model in here. In blue, what we can see are certain types of cells in the heart that we're going to call autorhythmic cells. Sometimes they go by the name pacemaker cells because these are the cells that determine how fast the heart beats. These are not the cells that do the physical contraction. Those are going to be what we call myocardial cells. I'll write this a little better in just a second. Myocardial cells are what you see. Those are the red cells that make up the heart wall. They're the ones that do the contraction. They make up 99% of the cells in the heart. It's only those pacemaker cells or those autorhythmic cells that send this electrical current through the walls of the heart. And if you've ever been in one of those, like those physical therapy rooms where they have those, whatever those pads that send electrical impulses, you can put them on a muscle and send electrical impulses to a muscle and the muscle contracts. So muscles respond to electrical currents. These cells send electrical currents, and it's going to be these cells that send electrical current through the muscle fibers that again tell that muscle fiber when to contract. You don't do this, but you would see the same thing if you stuck a paper clip into the light socket. That electricity would cause your muscles to contract. So what we need to set up right here is how is it that certain cells can respond to an electrical current? What is it about those cells that allows them to send an electrical current? And for that, we need to talk 1406 for a second. And back to this drawing. Just talking about electrical current. When we're talking about cells, it's going to have to do with particular ions moving into or out of the cell. And I'll just use this while it's still kind of congested real quick. The terms that I want you to see, the first term is this term depolarization. It's a term that we use in physiology to describe a cell taking on a bunch of positive charges. So the green and the blue arrows here are indicating a cell taking on some positive charges. The positive charges are moving from the extracellular fluid outside of the cell across that plasma membrane into this intracellular fluid. So that's depolarization. The intracellular fluid is becoming more positively charged. Or if we were to say repolarization, repolarization is the opposite. That's when a cell is basically purging itself of positive charges. So all of the purple arrows are showing you potassium ions that are leaving. And that's kind of right off the bat. I don't want you to get too worried about whether it's sodium or whether it's calcium or whether it's potassium. We will see a cell take on positive charges if we want the cytoplasm to become more positively charged, and we will see that cell get rid of positive charges if it wants to leave behind more negatively charged cytoplasm. So these ions, like we mentioned, they're all charged particles, so they can't just cross this plasma membrane anywhere. There needs to be certain channels that are going to allow these ions to cross. Right now I haven't labeled any of these names of these channels. The other thing that you can see here, these funny little shapes that I have drawn in the middle of this cell, those are just representing proteins that were made by this cell. Proteins are big, they can't cross the plasma membrane, so they're stuck inside that cell. And these proteins have a bunch of negative charges attached to them, a bunch of anions. So that's the reason these negatively charged proteins inside the cell is the reason if we just took a voltmeter and stuck it onto this cell, it would read in the negative millivolts, somewhere around negative 70 millivolts. So not all cells do this, only cells that can respond to electrical current. So we'll see muscle cells, we'll see nerve cells. They have this charge kind of built into them, and they can change that charge by taking out a bunch of positive ions or getting rid of a bunch of positive ions. We're going to graph these cells becoming more positively charged and then more negatively charged. And it's not that a cell, it takes on positive charges to become more positively charged, but instead of taking on a bunch of negative charges to become more neutral, it just gets rid of positive charges. So depolarization, taking on positive charges. Repolarization, just getting rid of positive charges. Let me do something real quick that's just going to make that a little less congested. I just want to talk about real quick why those ions would cross the membrane. So this again is kind of a detailed example of something that we would maybe see in 1406. Let's put that plasma membrane back up again. And this plasma membrane, before we add anything in there, what we're going to see, I'll just add some channels. There are going to be what we call leak channels. There are leak channels that are going to be permeable to sodium ions and also permeable to potassium ions. So there's a leak channel for both of those. I'll just draw these leak channels. Those were those hollow structures that I was drawing. These leak channels are always open. And ions are just going to be able to cross them in whichever direction that they want to cross. So just to label this, ECF, extracellular fluid. ICF is our intracellular fluid. And let's put those negatively charged proteins inside this cell. Maybe one more down there. Okay, that's fine. So now we can see, due to these negatively charged proteins, the cell is already measuring at about negative 70 millivolts just at rest. The other thing that I want to add in there, these leak channels, they're going to let ions move in whichever direction they want to move. So if these were the only channels that were around, we would see the same number of potassium ions inside the cell as we see outside the cell. And we got 4 of them there. So we're at equilibrium with respect to potassium ions. And we would be at equilibrium with respect to sodium ions. We'll put 4 of them out here. And now I'll have to put 4 of them in here. What I've just drawn right here with equal numbers of sodium and potassium inside and outside of the cell, we're drawing a cell that's going to have no membrane potential. It's going to be measuring somewhere on 0 membrane potential. But cells are not going to be at equilibrium like this because of another protein that we have to add in here that we're going to call the sodium-potassium pump. There's a lot of these sodium-potassium pumps. What these sodium-potassium pumps do, what's kind of interesting about those is that they take about 95% of the ATP that your body manufactures from whatever you ate for breakfast this morning. 95% of that energy is going to go to fueling these sodium-potassium pumps. What they do is they are constantly taking 3 sodium ions and throwing them out, and at the same time they're going to grab 2 potassium ions and bring them into the cell. So they're constantly getting rid of sodium. 3 of those sodiums go out, and they're bringing in potassium. So if we let these sodium-potassium pumps do their job very quickly, let's get rid of 3 of these sodiums. Those sodiums now go out here. And we've pulled 2 potassium ions from out here, and we're putting those inside. And that's just one turn of this sodium-potassium pump. It's due to those sodium-potassium pumps that we have an imbalance with a bunch of sodium outside and a bunch of potassium inside. Are we doing okay so far? All right, let's add a layer to that. The next layer is going to be what we call a voltage-gated sodium channel. And I'm going to also introduce a voltage-gated potassium channel. These are channels, and they open and close based on a particular voltage. So the first thing that's going to happen, we'll see, I'll just draw this in here as a little rectangle here. These voltage-gated sodium channels, when these open, sodium can actually move in either direction that it wants to when these things open, but sodium is going to move into the cell. I'm going to erase these and draw them back, but we'll see anytime that we're talking about a depolarization, it's typically sodium ions at some point that are moving into the cell. Any positive ions moving into the cell is depolarization. My question here is that there's 2 reasons, and let's see if we can pick these out. There's 2 reasons why sodium would move into this cell when these voltage gates open. Can anybody give me one of those 2 things? He's saying the uneven amount of sodium, there's more sodium outside than there is inside, so the concentration gradient, the fact that there's more outside than there is inside, is going to make it flow from high to low concentration. It's going to move down its concentration gradient into the cell. That's right, that's one of the reasons that sodium would rush in, but there's another reason that sodium would want to rush in, and that's that inside the cell there's a bunch of these negatively charged proteins. Positive charges are attracted. So not only is it moving down its concentration gradient, but it's also being pulled towards these negatively charged proteins. So when we see sodium ion get a chance to move into the cell, it's going to move in very quickly because there's 2 reasons for it to move in. Concentration gradient and change in charge, attracted to those anions. Okay, so what we just described there is depolarization. We just described sodium ions rushing into this cell, and the cell's taking on a bunch of positive charges. There's our depolarization. And now, without graphing this, let's just flip real quick. And instead of thinking about sodium at all, because to tell you the truth, sodium rushes in and it makes, for just a fraction of a second, there's a lot of sodium inside the cell. There's still a lot of sodium outside the cell, so we kind of lose that concentration gradient with sodium. It rushes in. There's going to be about equal concentration of sodium outside the cell as there is inside the cell. So in order to repolarize, what I'm basically trying to say is that sodium cannot turn around and leave as fast as it rushed in. So if we're going to repolarize the cell, getting rid of sodium is not the way to do it. Instead, there's another positively charged ion that we have built up with the concentration gradient. Just like you mentioned, potassium ions are in very high concentration inside the cell. There's not very many of them outside the cell. So when these voltage-gated potassium channels open up, if these things open up, well, we have a concentration gradient where potassium would want to leave the cell and go down its concentration gradient. But if you notice, potassium, it's going to have to leave those negative charges. It likes those negative charges. So we're just saying that in order for a cell to repolarize, the only thing that's pushing potassium out of the cell is its concentration gradient. It doesn't have that additional reason for the being attracted to the charges. It's the opposite for potassium. It's reluctant to leave those negative charges. And the reason I'm saying that is because if you have ever heard of a situation called hyperkalemia, we had this conversation with somebody in the physiology class that was talking about they watched a heart surgery and they gave a bunch of potassium to the tissue to stop the heart. And this actually happens in people that are hyperkalemic. Your neurons will stop sending signals. And what you need for a neuron to send a signal, neurons have to depolarize and then they have to reset. They have to repolarize, so reset and repolarize are kind of the same thing. And if a neuron can't repolarize, then it can't send another signal. It's a way to stop neurons in their tracks. So that's why this diagram had gotten so crowded a second ago, because at the end of the last class, I put a bunch of potassium ions out here in the extracellular fluid just to mimic us kind of injecting a bunch of potassium ions, or if you're hyperkalemic, maybe you get these from the diet or whatever it may be. In the case of hyperkalemia, you have so many potassium ions in this extracellular fluid that it makes it very difficult for repolarization to occur. Because if these voltage-gated channels open, potassium doesn't care. You've lost that concentration gradient. It won't leave the cell. If neurons can't repolarize, if they can't do this part, then they're not going to be able to reset and send another signal. So I guess we are just about ready to graph this depolarization where the cell takes on positive charges, and this repolarization where it basically purges itself of positive charges. We doing okay so far? You didn't think you were going to get any A&P lecture this time, but here we are. I know. Let's try this with better handwriting. Just do the whole thing again. And you know, I think it's helpful because in 1406, I was talking about the fact that you've got proteins, and proteins do a bunch of different functions around the cell, but we didn't really talk about specific proteins and specifically what they do in cells. We get to see some examples of that today. Let's get this out of the way. I'll leave that. Well, I'll draw the same thing. I just think it's easier to do it one step at a time. So what we were looking at over here are what we're seeing on that slide as the blue cells. On this heart model, they're using white fibers to represent these autorhythmic cells. These are also just called pacemaker cells. And we can graph their activity. I'll just squeeze it in right here. So we'll put time in milliseconds. And we'll put membrane potential over here. And membrane potential is always in millivolts. So we're really just measuring the charge of the cell. And we're going to see it go anywhere from above or below 0. I'm going to make this one up to plus 20. I'll put negative 20 down here. We really need to pay attention to negative 40, negative 50, and negative 60. I left some room below that, but we don't really need to know below that. It's important to make a dotted line at negative 40, because in these autorhythmic cells, that is what constitutes threshold. Let's explain threshold for a second. In order to do that, let's graph this activity of the cell. And I will say what's interesting about these autorhythmic cells, you might have noticed that earlier in the diagram, is that they're the only cell in the heart that is never at rest. It is constantly drifting between repolarization and depolarization. Repolarization and depolarization. So since there isn't any rest point, we're just going to start in the middle of one of these repolarizations. As soon as this cell gets into the positive range, that's about its limit. And it's going to start then repolarizing. So there's a couple of ways we can say that. When the line goes down, that tells us that the cell is losing positive charges. Its cytoplasm is becoming more negatively charged. So repolarization. And we know what's happening there is potassium ions are moving out of the cell. So the cell's becoming more negatively charged all the way down to negative 60. Then something happens at negative 60. Something kind of funny. And what's funny about it is that there is a channel down there that actually gets the name funny channel. It doesn't tell jokes or anything, but what is funny about this channel is that it opens in response to a negative charge. So as soon as this cell hits negative 60, that's the trigger for this funny channel to open. It opens at negative 60, and when it opens, for the same reason that you just were telling me before I crowded this drawing up, sodium ions rush into the cell as soon as it opens. And with sodium ions moving into the cell, that means the cytoplasm is becoming more positively charged. We would draw that line going up. Too much? All right, I know. All right, have a good day. So what's funny about these channels, like I said, they open in response to a negative charge. When they open, sodium ions are moving into the cell. They continue to move into the cell all the way until we hit negative 50. At negative 50, these funny channels close. Maybe I can just, well, I don't want to crowd too much in there. I'll just say me switching colors is indicating that these funny channels are going to close at negative 50, and instead another channel is going to open up. From negative 50 to negative 40, this channel that opens up is called a T-type calcium channel. There's my calcium ion. So T-type calcium channel. T-type just means transient type, meaning that it's going to be open for just a brief period of time. So between negative 50 and negative 40 millivolts, these T-type calcium channels are open. And when they open, calcium ions are moving into the cell. I'll just put this in here. Calcium ions into the cell. And if the cell is taking on calcium ion, any positive charge that the cell is taking on, that's going to make us more positively charged. We graph that by this rising line. T-type calcium channels close at negative 40, but there is another channel that opens up and takes us all the way up into this positive range. So between negative 40 and the positive range, this type of channel is going to be, I'll just maybe squeeze it right, I'll put it up here. We call this a L-type calcium channel. L-type just means long lasting. So these are going to be open for a longer period of time. When this L-type calcium channel is open, calcium ions are moving into the cell. So again, positive charge is moving in, the cell is depolarizing up until the peak, at which point this L-type calcium channel, it was open at -40, and it closes as soon as we get to that plus charge. So no more calcium ions are going to be able to move into the cell because the gate closes at the positive charge, and instead what happens at the positive charge is our voltage-gated potassium channel opens. So this is our normal voltage-gated potassium channel. When this is open, potassium ions are moving out of the cell. And as those positive charges are leaving the cell, like we were just graphing over here, it's leaving behind just the cytoplasm with these negative charges in it. So repolarization, potassium ions are leaving. It's going to continue to repolarize until we hit that magical number of -60. That again, -60 is the trigger for those funny channels to open. They're going to stay open all the way up until -50, at which point it's those T-type calcium channels that take us all the way up to threshold, -40. And then it's the job of those L-type calcium channels to take us all the way up to the positive range. Then once that positive range is reached, those voltage-gated potassium channels open and we're right back down to -60. So you can see these pacemaker cells are never at rest. They're constantly fluctuating between depolarization and repolarization. You also get an idea of how calcium ions are important in cardiac physiology. If you don't have enough free calcium ions in the bloodstream, it's going to be hard to generate those electrical currents. It also shows calcium can mimic, let me say that again, caffeine can mimic the effects of calcium in the heart. So it's one of the things that can affect heart rate. We had a graph over here. These cells are not doing any kind of contraction, but it's these cells that are responsible for determining how many times the contracting cells actually contract. So maybe with this color right here, something magic happens right there, right when we cross threshold. That crossing threshold is what is going to send a signal. I wish I could, without crowding that up, let's just imagine that this little asterisk is referring to a signal that's going to be sent over here to tell these other cells, these contracting cells, to actually contract. So depending on how many of these times we cross threshold, how frequently, that's going to affect how many times these contracting cells contract. I had highlighted, I'll go ahead and put this up here because we'll mention it. It's really this part of the graph that can be fine-tuned. This part of the graph is what we call pacemaker potential. This pacemaker potential can be influenced by the sympathetic or parasympathetic nervous system. Sympathetic nervous system is fight or flight, so it gets the heart rate increased. So what it does basically is it shortens the amount of time that you spend in pacemaker potential. It makes you reach threshold much more quickly. Parasympathetic nervous system basically inhibits funny channels in T-type calcium channels. Makes it harder for ions to cross. So it basically drags out how long it takes you to finally drift up to threshold. That can slow down how many beats per minute your heart goes through. Or by shortening that pacemaker potential, you can have more rapid heartbeats. But however many times we cross threshold over here is going to correlate to how many times these contracting cells actually contract. So let's talk about these myocardial cells. These myocardial cells are kind of described, they've got muscle right there in their name. So these are also called contracting cells. And let's graph these as well. We're going to do it the same way. I'll try not to let this get as low down on the screen because I know the monitor there is going to cut me off. We've got membrane potential and time in milliseconds. So time is still down there in milliseconds, but the membrane potential on the myocardial cells is going to be a little different. I'll put us going all the way up to plus 30. I'll somewhere sandwich in there 0 membrane potential. We can go down to negative 30. I'm going to put negative 70 in there, and then the other one we need to remember is negative 90 millivolts. And what's really going on there, negative 70 is the threshold value for these contracting cells. And at rest, these cells are actually at rest. We didn't see that in the pacemaker cells, but these contracting cells, we will see there are times in which these myocardial cells are not contracting, they are at rest. And that is just going to show up as a flat line. We will call that our resting membrane potential. I'll abbreviate resting membrane potential. These things rest at negative 90 millivolts, and they stay at rest until this little asterisk things happen. That is the signal that now is basically representative of, I think I used that analogy of a wave of people in like a stadium where they stand up and you see that wave kind of move around. Crossing threshold is going to send a wave of depolarization through the atria and then through the ventricles. And that wave of electrical charge is what's going to change. It's going to wake these cells up from resting and send them immediately into depolarization. These cells are going to quickly go from negative 90 all the way up to plus 30. So from negative 90 all the way up to plus 30. And what's going on here to make them so positive so quickly is what we just introduced over here, the voltage-gated, check mark it, I guess, the voltage-gated sodium channel. I'll abbreviate voltage-gated sodium channel. These are open, and this allows sodium ions to move into the cell. So rapid depolarization with sodium ions moving into the cell. This rapid depolarization comes to an end with another short little phase. I'm going to draw from about plus 30 down to about plus 10. I know in my graph I'm kind of close to neutral. But just for a little bit of time, there's a short repolarization where the cell becomes more negatively charged, just for a brief period of time. What's happening during that segment, there is what we call a T-type potassium channel. Again, T-type is transient. It's only open for a short period of time. And it's permeable to potassium ions. So what's happening in that little segment, we have potassium ions moving out of the cell. Hopefully that's not too congested there. In fact, let me make that more linear. So T-type potassium channels are open. Crowd that in there, and when they are open, potassium ions leave the cell. That's what causes this brief repolarization, but that flattens out. We're going to see right after this brief repolarization, we hit this flat phase that we call a plateau phase. And there's two things occurring in this plateau phase. The first thing we need to know is calcium. There are calcium channels that open. Calcium channels open, that's one. The second thing is that calcium ions are going to move into the cell. If you just remember what we just said, potassium ions during this little phase start to leave the cell. The cell is purging itself of positive charges. It's starting to repolarize. But as positive charges are leaving, now in the plateau phase, we have positive charges in the form of calcium moving in. I'll add, at the same time that calcium ions are moving into the cell, we still have these potassium ions moving out of the cell. So this plateau phase is, the reason that we're not changing any charge is because you have positive ions moving out in the form of potassium, and then kind of matching that are positive charges moving in the form of calcium. So we hit this little plateau phase. If you've had anatomy and physiology before, you might be thinking calcium ions moving into a muscle cell, that means contraction is about to take place, which it's about to. So let me pause on that for just a second and finish the graph. which is in a purple color. At the end of this plateau phase, what happens right here at the end of the plateau phase is those calcium channels close. So no more calcium can move into the cell. Instead, the only gate that's open are the potassium gates. These are the voltage-gated potassium channels. These voltage-gated potassium channels open. Potassium ions are moving out of the cell. And as the cell purges itself of those positive charges, it's becoming more and more negatively charged, all the way down to negative 90, where it remains at rest again until the next pacemaker cell sends a signal. And that electrical current ripples through these myocardial cells, and muscle cells are responsive to electrical current. It sends them into this immediate contraction. This, however, is not a graph of contraction. This isn't a graph that shows muscle contraction at all. Instead, this is showing the electrical event that precedes contraction. Here's where I just need to show one slide real quick. I'm just taking a step back to see if I left anything off. Let me pull this graph down real quick. We have drawn electrical activities on here. And in the A&P class, I always try and burn into everybody's brain that mechanical events follow electrical events. Again, going back to that little stem machine that I was talking about, where you can hook yourself up to those electrodes that send electrical currents, and when your muscle is hit with an electrical current, it'll contract. The muscle doesn't just contract on its own unless there's an electrical event. So mechanical events always follow electrical events. Or you can say electrical events precede mechanical events. And here's the graph of what we're looking at now that we've drawn all of these. The yellow stuff is just the pacemaker cells. Those are the autorhythmic cells that are moving through the heart tissue. And then the heart wall itself, all the red stuff, these are the myocardial cells that do the contraction. And these autorhythmic cells send a wave of electrical current through the muscle cells, which is what we've just graphed. And there is part of that electrical signal in which a little bit of calcium moves into those muscle cells. That is the thing that should be triggering some antenna to pop up in your head if you've ever talked about muscle contractions or muscle physiology. Let me just take a second to explain this graph. It looks a little more confusing than the one that I drew on the board, and maybe the only thing that we didn't talk about in our discussion are these little markings here. What is going on here? Just starting down here at the bottom, that arrow up or down is just telling you increased or decreased. That uppercase P is just telling us that represents permeability. How easy is it for that particular ion to cross the membrane? So just to show you, at this point in the graph, at negative 60, the permeability of sodium ions is really high because those funny channels are open. That's what all those abbreviations mean. Funny channels are open. The permeability of sodium is through the roof at negative 60, and that's why sodium moves into the cell. You can see the down arrow is representing that at negative 60, the permeability to potassium ions closes off. So no more potassiums move in at negative 60. Instead, sodium ions are moving in. Sodium ions move in until we reach plus 50. At that point, those funny channels close off. So permeability of sodium drops off. But at the same time, the permeability of calcium ions through those T-type channels is through the roof. So calcium ions come in through this part of the channel. And then as soon as we cross threshold, it's those L-type calcium channels that let most of the calcium through. Then those L-type calcium channels close off at the peak, and permeability of the potassium ions open up. Again, voltage-gated potassium channels are opened on this side, so potassium is moving out of the cell. So that's all those little things mean. It's talking about permeability of particular ions and whether or not those channels are opened or closed. I tried to make a comparison of both of these two things, and here is our more detailed example of what we just drew. Again, as soon as we come off of this resting phase, the permeability of sodium ions is through the roof. It's sodium moving into the cell that depolarizes all the way up to +30. And then for a brief period of time, potassium leaves the cell. That causes this short repolarization. And then as potassium leaves, some calcium starts to move in. So that neutralizes, the positive charges moving in and out of the cell kind of balance one another. So we hit this little flat plateau phase. Then the calcium channels close. You can see permeability of calcium drops off, but the permeability of potassium is still sky high. So now the only thing moving is potassium out of the cell all the way back down to -90. There's also this way of seeing it. I like this graph because it shows what's going on with the ion movement at different phases. During depolarization, we can see sodium rushing into the cell. During that brief repolarization, that's due to potassium ions leaving. The plateau phase, there's a mix of calcium moving in and potassium moving out. So those charges kind of balance out. We're flat for a second. And then during the repolarization phase, the only ion moving is potassium leaving the cell. So there's our repolarization. The thing that I kept wanting us to think about is this flat phase where calcium ions are moving into these contracting cells. The thing that you might be thinking about there, and again, here's that, what causes these cells to depolarize is the pacemaker cell depolarizing. These control the contracting cells. Here is this graph that is showing one of our contracting cells. And right there on that plateau phase, this part of the phase where just a little bit of calcium ions move into the cell. It turns out for cardiac muscle tissue, just a little bit of calcium ion moving into the cell from that extracellular fluid is all it takes to release a burst of calcium ions from inside the muscle tissue. Here's a muscle model that I grabbed. It's a skeletal muscle, but you also see these things in cardiac muscle. This little webbing thing that we see here, this is a chamber that stores calcium ions. There's a bunch of them all packed into this sarcoplasmic reticulum. This box right here is supposed to represent our sarcoplasmic reticulum that has a bunch of calcium ions stored up. And what is going to trigger the release of a whole flood of calcium ions out into this cardiac cell is just the trickle of a few calcium ions in. This is what's known as calcium-induced calcium release. Calcium-induced calcium release. So a whole burst of calcium ions can be released if just a few of them move into the cell. So very quickly, we see the cardiac muscle cells are going to get flooded with calcium ions. Now, the rest of this flow chart might not be familiar to you if you hadn't had the first part of anatomy and physiology recently. Calcium ions, whenever they are in the fluid of a muscle cell, they're going to lead to cross bridges that form between these myofilaments, actin and myosin. If that's not familiar, let me just take 1 second and pull up this graph right here. This is a close-up of a neuromuscular junction. It's a neuron that's talking to a muscle cell. And what you can see on the surface of this muscle cell, there is a smooth little plasma membrane. And then right there on the plasma membrane, there's a little pit that drops down into the interior part of this muscle tissue. If you look on these models, you can see these little holes or little pits on the side of this model. Those are called T-tubules, and they allow electrical currents that move across the surface of the muscle cell to get down into the interior of the muscle cell. What we find on the interior of those muscle cells, that webbing right there, that's the sarcoplasmic reticulum that stores a bunch of those calcium ions. And what we know about that sarcoplasmic reticulum is that it has voltage gates all along its length. They respond to an electrical charge. If an electrical charge goes into those T-tubules, it opens up these calcium gates. Calciums flood into this sarcoplasmic reticulum. Now the story about, here's all that calcium leaving that sarcoplasmic reticulum and flooding into the muscle cell. What's important about calcium ions moving into this muscle cell is they have to play a role in muscle contraction. And maybe I'll get somebody to help me demonstrate this. What we're about to do, right now what we're looking at is a relaxed muscle because this myosin filament is prevented from binding with actin. You can see the blue actin it has those little pits on it, those little dark spots. Those are what we call the active site on actin, and it's right there in those little pits that this myosin head wants to bind to. But right now you can see that myosin head cannot bind to any of those active sites because all of those active sites are covered up by that thin rope-like protein called tropomyosin. You can also see that round protein called troponin. That thing is holding on to tropomyosin. I think of those two characters as that Charlie Brown character that's always dragging around that blanket. Y'all know which one I'm talking about? That troponin, that's the Charlie Brown character that's dragging around the blanket. And the blanket is this tropomyosin. Carries it around everywhere it goes. So in a relaxed muscle like we see, there's 3 proteins involved. There's troponin and tropomyosin. They're always paired together. And the third protein is actin. Watch what happens when calcium ions move onto the scene, though. If calcium ions are present, calcium binds to that round little protein called troponin. And when troponin is attached to calcium, it actually it carries that blanket with it. And the blanket now has moved off of those active sites. They're now exposed and make it possible for actin and myosin to form a cross bridge. This is when you get muscle contraction, when actin and myosin can form these bridges. But before calcium ions show up, that tropomyosin rope is blocking all of those active sites. Let's try this. I am going to be troponin. I'm going to be that rounded protein. Troponin, kind of like me, has 2 hands, and troponin doesn't want its hands to be empty. I'm going to ask y'all to participate because you have a nice emblem on your shirt. Just come up here and let's face everybody else. That, or even better, we're going to use this patch right here on his shoulder. This is going to be the active site. Ethan is going to be, you are actin, and that patch is your little active site. And that patch is where the myosin head would want to bind to. Okay, I'm going to be troponin, and Tyler's going to be my tropomyosin rope. Okay, so I'm carrying him around wherever I go, and I don't want my other hand to be empty. So in a relaxed muscle, I've got one hand on actin, I've got one hand on tropomyosin, and I'm holding tropomyosin so close to actin. Just put your hand out here and cover up that active site. So right now that active site is covered up. Nothing can bind to it. We're guaranteed to have a relaxed muscle. But Brandon, would you walk over here? You're going to represent calcium ions. So as you leave that sarcoplasmic reticulum and move into the muscle, I prefer to hold on to calcium ions, and I'm going to have to let go of something. I'm not going to let go of tropomyosin. I'm going to let go of actin. And if you'll just take a couple of steps back and stand right there. Now I'm holding on to calcium ions because they're present. And as long as calcium ions are present, we've got that exposed active site. So now cross bridges can form. We can get a contracted muscle as long as calcium is distracting me. But as soon as the calcium goes back into the sarcoplasmic reticulum, I don't want my hand to be empty, so I'm going to come and grab back onto actin, and that active site is covered up. So now we got a relaxed muscle. Thank you all very much for that. So whether the muscle is contracted or whether it's relaxed has everything to do with the presence or absence of calcium. That's all that we're showing here. Back to the cardiac physiology PowerPoint. We were just right here, and I was just talking about how a little bit of calcium moving into the cell triggers a burst of calcium to leave that sarcoplasmic reticulum. So that was when calcium was moving over here into the muscle cell from the sarcoplasmic reticulum. It's going to lead to muscle contraction. And if we want to graph how strong a muscle contraction is in the cardiac cycle, we see the contraction, this blue line, the forceful contraction of the muscle starts right when the plateau phase starts. At the beginning of this plateau phase, that's when the first calcium ions start to move into this cell. And with a little bit of calcium ions, there's a little bit of a contraction. And as we get more calcium ions, eventually the maximum number of calcium ions right towards the end of that plateau phase, that corresponds with the maximum strength of contraction in the heart muscle. People that are having these heart attacks where the heart is too contracted, they want to prevent the heart from contracting. They give them these calcium blockers. And that's preventing calcium from moving in through this plateau phase, trying to prevent a prolonged contraction. You can see when we get rid of calcium ions at the end of that plateau phase, that corresponds with the relaxation of the muscle. So we can see in red here the electrical events that always precede the mechanical events, which are in blue. That contraction would not have just happened on its own. This electrical event is what started that mechanical event. And again, it started with calcium ions moving in at that plateau phase. You know what? For the purposes of this class, I am going to hold off on this EKG because I can just explain that to the A&P people. This is basically the same thing that we were just talking about. The waves are electrical events and the flat lines are mechanical events. And it shows that electrical events always precede these mechanical events. This is what we call the cardiac cycle. I like this picture. This is a moving picture of the cardiac cycle. What's cool about it is it shows you that both atria contract at the same time, and then both ventricles contract at the same time. That's one cool thing about it. I also like that it shows you the atria contract right before the ventricles contract. That's another cool thing about it. And the other thing I like about it is it shows that when both chambers are relaxed, notice when you watch this, there's a fraction of a second where both chambers are relaxed. And during that time, when both chambers are relaxed, that's when the ventricles are filling with most of their blood. The ventricles fill passively. Just there's blood pressure in the vessels, and it's that pressure of the blood that fills up these ventricles almost 70% of the way through. And then what pumps the last 30% of the volume into the ventricles, that's when both atria contract. So the ventricles fill almost all the way up, then the atria contract and force the last bit of blood into the ventricles. And then you see for a fraction of a second as the ventricles contract, the first thing that happens as those ventricles start to contract, here it goes, boom, those AV valves close off. See how those close before these semilunar valves open. The closing of those AV valves, these valves are like trap doors. They open easily in one direction, but they don't open in the opposite direction. They slam shut. So when the ventricles start to contract, those AV valves that get slammed shut, that makes the first sound of the heart, the lub sound, closing of the AV valves. The ventricles, as they continue to contract, the pressure builds so much that those semilunar valves open. Notice how those open. And then watch what happens right as the ventricles start to relax. The first thing that happens as they relax, those semilunar valves are going to close. Watch right here. Boom, those close first. Those close, and then these open. The closing of those semilunar valves, the slamming shut of those semilunar valves, is the dub sound of the heart, the second part. So the sounds of the heart are made by closing valves. The first, the lub sound, is the closing of the AV valves, and the second, the dub sound, is the closing of the semilunar valves. That's the cardiac cycle. That's all that we were graphing with this. This just shows electrical events preceding mechanical events. Depolarization of the atria precedes contraction of the atria. Here we see depolarization of the ventricles, and following depolarization of the ventricles contract. So mechanical events are always following electrical events. Here's the last thing that I'll show you, just jump into some details. I mentioned that sympathetic, we'll just look at it this way. Sympathetic and parasympathetic nervous system can affect heart rate. So above this dotted line, this is just representing how many signals are sent from the pacemaker cells to these contracting cells. This number of signals will get these contracting cells to contract, looks like 4 beats. Down here, we've got 6 beats squeezed into the same amount of time. And the only difference between the top line and the bottom line is the amount of time spent in this pacemaker potential. I said sympathetic and parasympathetic nervous system can affect that part of the graph, the pacemaker potential, because what's down there are those funny channels and those T-type calcium channels. It turns out that parasympathetic stimulus can basically block those T-type calcium channels and the funny channels. It makes it more difficult for ions to move in. And that's the reason why we see during parasympathetic stimulus, I'll just go back to this graph. During parasympathetic stimulus, it takes longer for us to drift up to threshold, so this slows down heart rate. Sympathetic stimulus looks something like this, where under sympathetic activity, we can see those T-type calcium channels and funny channels opened even more widely. They're stretched open, so ions move in even more quickly. And that's what makes it possible for us to reach threshold even more quickly. So sympathetic and parasympathetic nervous system really just affect pacemaker potential, funny channels, and T-type calcium channels. What else? I'll just end with this. I started talking about my heart, how that when my heart rate jumped quickly from 90 beats to about 220 beats per minute, the chambers were beating so fast that they weren't pumping any blood because the chambers didn't have time to fill up. So heart rate isn't the whole story when you're thinking about how much blood the heart pumps. That refers to cardiac output, which is heart rate plus stroke volume. You can see the abbreviation of it here. It's described here as just the volume of blood that the ventricles are pumping each minute. So that has to do with how many times the ventricles pump, but also how much volume of blood is squeezed out per beat. And both of those play a role in cardiac output. The reason that we just want to see this graph is because I had mentioned sympathetic and parasympathetic activity can affect heart rate. And we can see there are branches of the parasympathetic nervous system that go directly to those pacemaker cells. So these are, notice how they're not touching any of the contracting cells of the heart. They're only touching the pacemaker cells of the heart. So parasympathetic nervous system can affect heart rate. But sympathetic nervous system, look at the green fibers. These also go to the pacemaker cells. So sympathetic nervous system can also affect heart rate, but in addition to heart rate, it has these branches that go to the myocardial wall. And there they can affect stroke volume, how forceful each beat is. If you've ever tried to wring water out of a dish towel, you know that getting that twisting motion, really kind of wringing it out, you're going to get more water that way. And activity of the sympathetic nervous system really increases how much of a pumping action there is on those ventricles. So it affects stroke volume as well as heart rate. For this reason, we can say the sympathetic nervous system can affect cardiac output more than parasympathetic nervous system. And also, I guess, additional knowledge for this. When we were talking about cardiac physiology, we mentioned that unlike your brain fuels itself only on glucose. So if you don't have enough sugar in your bloodstream, your body will have to make its own sugar by breaking down lipids or breaking down proteins. And it's called gluconeogenesis, making glucose, new glucose, from things that were not glucose, like lipids and proteins. The heart doesn't have time for that. The heart has to be able to use multiple energy sources because it never can take a moment off. So the nutrients available to the heart are glucose, as well as fatty acids and lactate, multiple energy sources, and it can easily shift between the two so that it never has to wait for energy. It is a needy organ, so not only does it need a lot of energy sources, it needs a lot of oxygen. It takes most of the oxygen from the blood, about 65%. And in our A&P class, we talked about the blood vessels around the heart having a particular receptor in them called beta-2 receptors. I know that doesn't ring any bells in here. Beta-2 receptors are going to be ones that dilate in response to sympathetic nervous system. So dilation of those blood vessels is what allows for fresh blood to hit the heart wall.